Organoid intelligence. What is that? Well, it turns out that organoid intelligence is using tiny little mini brains to work as like a sort of pseudo computer. But we'll get there. I was hoping to start my research process by watching all of these videos to get like a base understanding of how this works, because a lot of time when I'm researching things, I just spend a lot of time flailing around trying to understand the basics. So I was like, oh, maybe if I watch some YouTube videos, that will give me a basic understanding, and then I can like do more in-depth research after that. But what it did was just overwhelm me and I ended up flailing around for a long time trying to get purchase on this topic. So long story short, I started here with organoid intelligence. It's where I finally kind of got it together. Organoid intelligence may soon power our computers. So one of my first thoughts in reading this article is that it sounds like hype, like heavy hype, a future where computers are powered by lab-grown brain cells. But let me explain it in spite of the hype. So organoids are these three-dimensional tissue cultures that they usually get from human stem cells, human skin stem cells usually. And they're basically just a little clump of cells in a petri dish. If you remember from a previous video, pluripotent means young, like a young stem cell that hasn't gotten old and lost its ability to change into anything it wants. So apparently these organoids have been used to, they grew like 800,000 of these little clumps of brain cells in a lab and used them to perform a game, Pong. I guess in this video the brain cells are playing Pong, like they hooked them all up. One of the videos I watched was talking about how they like set them in little slots next to each other, and then they actually formed connections. They grew connections together, and then I'm not sure how they trained them exactly, but somehow they were able to like instigate learning in the little clumps of brain cells that were tied together, and they learned how to play Pong. But that was 800,000 of them, which sounds very labor intensive. I was struggling to read this whole article because it sounds very hype, and I don't know, I was struggling. So somebody give me organoid intelligence in a simple manner. Explain it simply, please. So that brought me here. Diving into organoid intelligence. You can see the little clumps of brain cells in the petri dish there. They're tiny. So I don't know if this was stated before, but they're using brain cells to enhance computer functionality. So it's not like they're going to be able to turn the brain cells into a computer themselves, but they can hook them up to a computer and make the computer work better, which I feel like is an important distinction. But apparently they've been doing this for almost 20 years, nearly 2 decades. Here's where they say they get the stem cells from human skin samples. Here's where they talk about the shortcomings of AI and traditional computing, because like AI is not very energy efficient. It doesn't learn very well. You can't teach it new skills easily. It doesn't do complex decision-making stuff. But the human brain is great at all of that kind of thing. You want someone to plan your trip to Mars? Ask a computer. You want someone to pick a face out of a crowd without millions of hours of data plugged in? Ask a human brain. Somebody in one of these articles said something about how like if you tried to teach a person to walk, if you tried to teach like a human child to recognize a picture of an animal, it would take like 10 different exposures to that animal for the child to begin recognizing it. But for the computer, it's like 10,000 images shown and it still is like recognizes it poorly, something like that. So at this point in my research process, I was thinking, okay, I have some main questions that should probably be answered. And the first one is, how do brain cells talk to each other? Like if these little brain clump cells are communicating, how exactly are they doing that? And how does growing things from stem cells actually work? Because I've talked about it several times on this channel and I still don't really understand exactly how things are grown from stem cells. So let's look into that. First of all, brain cells. And this is just kind of a testament to how difficult of a time I was having researching this topic and getting my brain to like focus. Is I had to go on the Frontiers for Young Minds and look at the kids one. So in your brain, neurons are doing the communicating, right? You get 80 billion of them, which is a lot of neurons. So they like to use the analogy of the tree. Here you've got the neuron, you got the nucleus in the middle, it's holding it all together. You've got the little branches that are coming off that are called the dendrites, and you've got the long root tail thing that's the axon coming down. Okay, and then you've got the synapses at the very tips of the root parts. So you can see in this image that the dendrites are the ones that are receiving things, and then the axon terminals, these little synapse parts right here, are the ones sending things. So the message gets sent down the axon and then received by the dendrite, and then it can go down this axon to that dendrite and so on. I feel like we shouldn't be teaching children this way. They actually use the wrong like terminology. They're just trying to say that the axon terminal is a terminal because like in an airport, like things are leaving the terminal and like going out into the world. But guess what also happens in terminals? People come in. Education these days, man. Anyways, so you've got these neurotransmitters in there. So I just wanted to double check that I'm correct in thinking that axon terminals only send things. So I went here. Axons, the cable transmission of neurons. And I found out that axons are teeny, teeny, tiny. They're just like a millimeter. Oh, this is a millimeter in length. Several times thinner than a human hair. So apparently, like some of them are pretty short, but the long ones can go from the brain down the spinal cord and can go for more than a meter, which is kind of crazy. I didn't realize that there was neurons that were that long or that big. So like the little synapses here are giving the neurotransmitters to the dendrites, but the gap is only 20 to 40 nanometers wide. Like, so they're not touching, but they are very, very close, 20 to 40 nanometers away from each other. And I was like, how big is that actually? 20 to 40 nanometers. So I looked it up and it gave me this image, which is not super helpful. I feel like going from DNA times 1,000 to bacteria to another 1,000 is like, okay, thank you for that helpful comparison. Not going from a strand of hair to a house. Like, who taught you guys about comparisons? These are very poor. Anyways, just all that to say that DNA is 2.5 nanometers across. So if this gap is 20 to 40, it's like several DNAs stacked end to end. That's a very small gap is what they're trying to say. So the way that these things are actually communicated is you've got electrical impulses, that are starting here, and they're traveling down the axon. And then they are, when they get to the synapses, they release the neurotransmitters, which are like little chemicals. So it's translated from an electrical impulse into a chemical impulse at the end. The dendrites pick up that message, that signal, those neurotransmitters, and read it. And then they communicate that to the cell body, which starts the process all over again in the axon, and away it goes. And here you've got the myelin sheath, which not all neurons have this myelin sheath, but this is kind of like a insulation to help the electrical signals travel farther. So if you have neurons that are going across parts of the brain, you'll get that myelin sheath so they can travel farther. But if it's just like local, they're just like talking to each other, neurons, then you don't usually get that myelin sheath on there. So if your axons start to get broken or damaged, then you end up with diseases or neurodegenerative diseases. Things like Alzheimer's disease or motor neuron or Parkinson's disease. So people have actually been studying this using roundworms. So interestingly, when an axon is damaged with a laser, it sends out a signal to the surrounding tissue to be cleaned up. It's like, hey, I'm not fit for use. I'm not fit for service anymore. I need you to take me out. So these little proteins will come and like hasten the degeneration of the axon. So they're hoping that if you can figure out a way to like prevent these proteins from like coming to clean up the damaged neurons, you might be able to slow the process of like these neurodegenerative diseases. But also, there's also like save me signals that get sent out to initiate nerve repair, which would be like super awesome if you could figure out a way to trigger that. They don't elaborate on this, which is kind of a bummer because it was interesting. It seems like they maybe haven't elaborated on it because they don't understand it that well yet, and they are still researching it, but I'm not sure. So they have this handy menu on the side over here that's like talking about all the different brain functions, and I just started jumping around because I was like, this is real interesting. So next place I went is what is a neuron here? What is a neuron? And so just like we talked about earlier, fundamental units of the brain and nervous system. Apparently the other one said 80 billion. This one says 100 billion neurons in your brain. And that's just like actual neurons. You also have something called glia, which we will talk about in a minute. And the creation of new neurons is called neurogenesis. Remember that for later. So you've got your dendrites here, but these little axon terminals right here. Oh, sorry. This green line is the axon terminal. And the blue line is the dendrite. And these little spines are what is actually picking up the chemical neurotransmitters. You can see them in real life right there, which is crazy. I don't even know what this measurement means. It's got to be like 1 nanometer or something. Very small amount. So we talked about these concepts and definitions, axons, dendrites. The spines are the little protrusions right here. They're sticking off of the dendrites. And the action potential, I think we might have mentioned, is the electrical event that travels down the axon and releases neurotransmitters into the synapse. Action potential, very important part of the process. So then I went to this part, types of neurons. I didn't really know this, but there are a bunch of different types of neurons, or I think four main types. So you've got sensory neurons that are activated by sensory input, like things like hot or cold stuff, or senses light, it'll be activated. And then most sensory neurons are pseudo unipolar. This one, pseudo unipolar, meaning they don't really have dendrites, it looks like they just have axons, which would actually make sense, because if this part of the cell is receiving like input like heat, it gets hot, it gets cold, it senses light, then it just sends out little stuff. It doesn't need to receive anything from other neurons, which makes sense. And then your motor neurons are the ones with the most like stereotypical neuron shape. These ones, the multipolar ones. These are the ones that are directly controlling all of your muscle movements. All right, we gotta keep moving. Onto glia. What are glia? It doesn't really say specifically on this page. It just says that glia are other cells in the brain that are like helper cells, but they look pretty similar to neurons, but they're not neurons. It used to be thought that they were just the housekeepers of the brain, but now we're learning new things about them. So I went here, obviously, types of glia. Let's learn more about them because it's interesting. And here you've got all the different glia that are helping out the neurons in the brain. Astrocytes, microglia, satellite cells. And there is a lot of interesting stuff in here, but I was kind of like, this is maybe a little bit too far off topic. So let's focus up a little bit. All right, so coming back to how neurons actually work. So you've got your neurotransmitters, you've got your action potential coming down the axon, and here's the synapse. And then so the action potential electrical impulse triggers all of these neurotransmitters to be like generated. I'm not sure where the chemicals are coming from in the synapse. How does it replenish its chemical stores? I'm not sure. Anyways, it releases these little chemicals into the void, the cleft, and then the dendrite spine picks it up and receives it, and then starts the process all over again in the next one. So apparently, like most neurons are actually monolingual, which means they can only release one type of chemical or neurotransmitter. So they can only communicate one thing, which is interesting. I had kind of assumed that they could do something kind of like DNA, where they had like 2 or 3 different options, and depending on what they were like affected by, they would release a different one. But apparently they can only like transmit one thing. But the good news is you have like 100 billion of them. So these are some of the neurotransmitters that are released from the synapses, things that are being like used to communicate across your brain. And there's 3 different types of neurotransmitters. Oh, sorry, getting into this part of the website. What are neurotransmitters? These little parts, chemicals. So there's 3 different types. There's excitatory ones, there's inhibitory ones, and there's modulatory ones. So obviously excitatory ones continue the process. They excite the neuron and like get the process to continue across the different neurons as it travels across the brain. And the inhibitory ones, where it's like, if you trigger this neuron, it will stop there and it will contain it and won't trigger the neurotransmitter release. And then the modulatory one, which is like a sort of a calming influence. It gets everything to calm down, or just like stop moving as much. Excitatory and inhibitory are like instantaneous ones. It's like as soon as the neuron receives that message, it stops doing what it's doing, or it starts doing what it's doing. But the modulatory one takes a lot longer to take effect. So at this point in the research process, I was like, I think I have enough information to kind of like go back to that original article and like get at least understand things enough to like start asking questions about what they're saying about brain organoids. But first I want to do the stem cells question, which is like how does growing things from stem cells work? So that brought me here, and I didn't have time to actually start reading this, so this is for next time. Be back in a flash. And like the flash, I am back. I realized right after I stopped recording yesterday that I forgot to talk about neurogenesis, which is just the ability to create new stem cells when you're an adult. Alright, so stem cells basics. What are they and why are they important? So we kind of already know a little bit about this from last time, but let's talk about it. So this paragraph was incredibly roundabout. If I hadn't already known what pluripotent stem cells was, I would have had no idea based on this paragraph. But pluripotent stem cells are young stem cells that haven't grown up yet. They're not the legal drinking age. They're still young enough to just turn into whatever cell in the body that they need to. It's like basically when you're an embryo in utero and you are trying to become a human, your cells are growing, and the stem cells can be like, oh, we need some blood cells over here. Oh, we need some bone cells over there. And they can just do all of them. But then adult stem cells don't have that ability anymore. They are unipotent, maybe? I'm not sure. But they can only turn into the kind of cells that are like in the surrounding tissue. They only have the ability to become a couple new cells. They can't just turn into whatever. These are in more specific anatomical locations, places like the bone marrow, the skin, a couple internal organs. An interesting thing about these is they serve as the internal repair system for the cells. So like if your cells are getting damaged in your body, these stem cells will start to like regenerate new cells that can take the place of the old cells. And the interesting thing about these is that when they divide, like when they go through the process of mitosis like a normal cell, they can either turn into two new stem cells, or they can divide into a stem cell and a differentiated cell, or they can both be differentiated cells. So this is kind of how they maintain themselves as like stem cells. They're not just like stem cell turning into two new like bone marrow cells. It's like stem cell turns into one stem cell, one bone marrow. Or it can do the two bone marrows, but usually it's like either one, or it can do two new stem cells. So it was actually in 2006 that they found out that they could reprogram the embryonic stem cells or the adult cells to be embryonic, which is a long time, almost 20 years ago already, that they've been reconditioning stem cells. Pretty crazy. Okay, so before I kept going, I was like, I really want to know, where are stem cells coming from, like exactly? Like I know they're coming from the bone marrow. I know like some of them are adults and some of them are young, but I want to know like where exactly are they coming from? Like there's a cell membrane, there's some tissue, like in that tissue there are different cells all over the place. Where are the stem cells? Like are they floating off into space? Where are they? And let me tell you, this was a rabbit hole, and not the good kind. So I searched up this, where exactly are adult stem cells found? And then I had to change it because I found nothing based on that. And I said, point out a stem cell to me in the body. And that brought me to this article, how blood stem cells are collected. And I was like, oh, goody, maybe they'll tell me. And then it was like, no, we use a needle to harvest your stem cells. And I was like, not what I wanted. So I came here, how are stem cells collected? And this told me that they're collected from the blood. And I guess bone marrow ones are better because they like to like siphon out the stem cells from the bone marrow and then put them into the blood because they're easier to collect from the blood, which again doesn't really tell me where. So I searched up, how are stem cells from skin harvested? Because I know they were talking about taking stem cells from skin samples. So that brought me here. Where do stem cells come from? Also useless. It just talked about like, oh, these are interesting places in the body where stem cells are found. Brain, blood, skin, bone marrow. Like, thanks, I already knew that. And then it talks about all the cool things that you can do with stem cells. I don't care. At this point, I was like, what the heck is going on? Why is this so difficult to find out? Like, am I asking the wrong question? Are stem cells called something else normally in like scientific research? And stem cell is like the weird layperson term that they use. So I came here, researchers turn skin cells into stem cells. I was like, okay, maybe this will help. Actually, but then I realized that would not actually help with like understanding where, but this would maybe help me understand like how they're actually reprogramming the cells, which is kind of an interesting topic, but not what I'm looking for. So I came here, how are stem cells harvested? And they gave me this quote. After your marrow is extracted, the stem cells are concentrated and isolated in our specialty lab, then removed from the collected sample while you wait in the comfort of our Huntsville, Alabama clinic. And I was like, yes, thank you. That's useful to know, but also it's not answering my question. So I searched up instead, stem cells and collected concentrated lab isolates. And that brought me here, isolate cells from blood. And this was kind of an interesting look at how they like separate the different cells from like blood, and they're using centrifuges to like separate the blood into like plasma, and I'm not sure what PBMCs are, and all these different things that just come from the blood naturally, but there's no stem cells in there, I think, if you'll notice. So I came here, stem cell separation technologies, which once again, they're talking about how to get stem cells out of places more easily and like using better techniques, which is awesome and great, but not the answer to my question. And at this point I was like, I'm over it. I've spent way too long trying to figure out this question. So I decided to ask Betsy, like, how can you tell me, or can you at least point me to resources? Like, can you help me out, Betsy? Because I am struggling. So this is my conversation with Betsy, and she pointed out a couple things, and she gave some articles to look at, but I was like, I don't know that I want to spend so much more time trying to find the answer to this. So I just looked up epithelial stem cells. I was like, maybe I can just get a picture of an epithelial stem cell. I can look at it in a diagram or a graph or something, and that'll be good. And that brought me here. And I see no stem cells on this picture. Correct me if I'm wrong. Oh, there's a libel stem cell. There are stem cells on here. How did I not see those before? I'm glad I saw that just now because I looked at this page. I was pretty tired after trying to like figure out where and reading a bunch of stuff, so I did not see these before. But there's the stem cells right there. I might need to come back and look at this more later. It's absolutely ridiculous that it took me this long to find this, but what can you do? Anyway, so I went back to the original article to learn a little bit more about stem cell basics. Okay, back to this one. Okay, here's where we left off. So they're not really sure at this point, like what is controlling the balance between like the three different options of stem cells that are being created. They just know it's happening. Down to here, how do you grow stem cells in a lab? Put them in a petri dish or a culture dish. Contains a nutrient broth, and then they will do the rabbit thing and attach and divide and spread over the surface. Cool fact, the original cells can yield millions of stem cells. So from that original batch of, I don't know how many they're seeding it with, like maybe 10? I have no idea, but that original little batch can yield millions. I'm wondering how they get them to start dividing, because I know that stem cells will can remain undifferentiated or like unsplit for a long time. So like if you just put them in the nutrient broth, do they just start dividing on their own, or do you need to is there a catalyst that's needed or what? So the next question is, how are organoids grown from stem cells? I'm assuming they're just going to put like a little tissue sample from something into the little clump of stem cells that has been grown and then let the magic happen. But I'm not sure. That's for tomorrow. So I'll be back to tell you about it. I'm back, baby. All right, we're looking into what are organoids and how are they made? Specifically, how are they made? Okay, so apparently there are two different things, organoids and spheroids, and they're different things, although very similar. And I'm not sure that I actually really understand the difference, but it seems like spheroids are like just primary regular cells that kind of like have been grown in 3D culture. And stem cells are assembled with stem cells. And that's the difference. So I guess like the spheroids are like used by like ordinary cells doing mitosis. They just put a clump of cells in there and then activate them, and then those cells just like start to split into themselves, and they're not changing it into anything else. They're just becoming more of that same cell, I guess. And that's how you get a spheroid. I'm not sure. I looked into it a little bit, and that brought me here. Like spheroid versus organoid, what's the difference? Spheroids are simple clusters, usually coming from tumors or embryos. They don't require scaffolding, and they simply stick together. So I'm not sure. I think so. Yeah, I'm assuming that they're just like grabbing bunches of cells and like putting them all together in the same petri dish, and then those cells that are like the same type of cells are just sticking together and forming these larger bundles of spheroids. I'm not sure, but I didn't feel like it was that relevant, so I stopped pursuing that angle. Organoids are similar in size and shape, but they are quite a bit different, if we're being honest. So how are organoids made? I don't know if I've talked about this much, but organoids really are kind of a cool idea because you can learn about organ development or like modeling diseases, modeling like how drugs affect certain organs and a bunch of stuff. They're really pretty cool, but that's not really a topic for today. I want to know how they're made. So I'm not going to lie, I was a little confused by this article. They kind of got very technical, it was like oscillating back and forth between like very technical and like a very layperson, and it was confusing. But I think what I got out of this was they put them in these little petri dishes and put in the culture or like the broth, the soup they need to survive, give them energy and whatnot. And then and that broth has to mimic the environment the cells would like naturally be in like the human body, naturally. And then after a while, they enzymatically detach them from the wells, like the feeder wells, I guess, feeder cell wells. I'm not sure what the wells are. They take the pluripotent colonies of stem cells and attach them to a different plate. And then over like a couple weeks, they start to form embryoid bodies. So at this point, they haven't really done anything other than just like cultivate the stem cell population. And then once they want to start like inducing them towards like tissue specific things, like they want to start growing a brain organoid, they want to start growing a lung organoid, whatever, they introduce tissue specific induction media, which I feel like is a very confusing way to say that, but that's just me. Like honestly, it's so confusing. I'm not even sure that I'm getting this right, but I'm hoping. And then after they've done that one time, they like start to develop a direction of where they're going to go. And then it takes several months of continuously culturing them with the tissue specific media. And then they can turn into things, intestinal ones, liver ones, kidney ones, eye and brain ones. So I was like, okay, that's cool. Let's see, how big are they? How big are organoids? And that brought me here. Organoids by design. So this says that they are Only usually they're only a few millimeters in size, so they were real tiny because there's not a lot of passive diffusion of oxygen and nutrients in the like culture solution. But what they can do to like kind of make them grow bigger is engraft them onto vascular beds in animals, which I'm not sure what a vascular, I don't understand the sentence, a vascular bed in an animal. So are they grafting them into an animal and then letting them grow in the animal? Or I don't know. I tried looking this up. I didn't find anything. So when they're doing that, they can grow into centimeter-sized tissues, which is significantly larger. And I was trying to look for a picture of like these organoids. Like it would be pretty cool to see a centimeter-sized organoid, but I couldn't find any pictures. So I was thinking like it seems like what's important for the organoids to get bigger is the vascular system. So I was like, let's look that up. Oh, also, they're trying to engineer vascular systems on their own so they can manually produce the organoids without having to graft them into animals. Anyways, what's vascular system? Brought me here, overview of the vascular system. And as I'm sure like all of you already know, vascular system is just the things in your body that carry blood or lymph fluid, arteries, veins, capillaries, all that jive. So is it blood that the organoids need? I'm not sure. I don't know how all that ties together with organoids, but that's what I have written down. All right, so I was thinking originally I would go back to the original article that we started out with, but then I was like, you know what? Now that I know a little bit more, this is 100% hype. I'm not going to read through this and try and get stuff because it's just going to take me in 1,000 different directions. That it's like, this is so cool. This is so cool. This is so cool. And I'm like, that's not going to get me anywhere. So I just searched up organoid intelligence again, and it brought me here. The new frontier in biocomputing. So this abstract made me feel like I would just want to read this article or this publication and just kind of like go through it point by point because it seems like it's going to be a pretty good time. Although I don't know if I'm going to look into this. They're thinking the development of these things could elucidate helpfulness in like degenerative diseases such as dementia and like help with therapeutic approaches. And like, I don't know that I'm super interested in that. I feel like, I mean, it's obviously cool, but I'm not going to do research on it. But everything else. Okay, so let's look at the intro. So they kind of start giving like justifications for why they're looking into this. Human brains are slower than machines at processing simple information, like math, like we talked about before, but they are way better at processing complex information if the data is not very certain or there's limited data. And they outperform computers in decision making on like big old heterogeneous, incomplete data sets. Brains are real good at learning how to walk. Computers are not. So then they get into like how much processing power it actually takes for like computers versus machines. And they spend a fair minute on this right here. Although they are throwing in a lot of sentences that are like very contextless. Like the world's fourth largest computer took 40 minutes to model 1 second of 1% of humans' brain activity. Like that sounds really cool, but I have absolutely no context for any of that. Also, the brain has a storage capacity of 2,500 terabytes. All right, so more on like power differences. They give the example of a larval zebrafish can like navigate the world successfully and not get eaten by hunters and predators. And it only uses 0.1 microwatts to power its tiny little brain. And an adult human will consume 100 watts. Doing what? Over what time period? I'm not sure. I'm hoping that they are citing these sources and like actually giving context for these numbers, but that's what they're saying. In contrast, state-of-the-art machine learning models typically operate at around 10 to the 6th power watts, which is a lot. The most powerful supercomputer reaching 1,100 petaflops. I don't know what petaflops are. The power consumption of the new supercomputer is 21 megawatts, while the human brain operates at an estimated 1 exaflop and consumes 20 watts. So they're just trying to highlight that like the human brain is like exponentially better at computing while efficiently using power, energy. And they give this table, this figure, which if you want to look at this figure later, it's like, once again, there's no context here, but the numbers are startling. If you want to pause the video and look at this, you can, but I'm going to move on. So then again, they talk about how humans learn simple tasks much more quickly using only around 10 training samples. For things like honeybees, which are like simple organisms, they need very few training samples to learn things. And then machines couldn't learn the distinctions with 10 to the 6th power samples. I don't know what that means. Do they just show like it's doing machine learning, I guess? Anyways, and they give the example of AlphaGo, which beats what's his face at Go, which is pretty incredible. But like if you think about how much time it took to train AlphaGo, it was 160,000 games, which is equivalent to a human playing for 5 hours a day continuously for 175 years. So I think they've made their point pretty well. Human brains are more efficient at learning these complex activities than machines. Currently, they like to make these statements. Human brains could theoretically meet the same US data storage capacity using only 1600 kilowatts of energy, which is kind of a crazy statement. But once again, like no context. I don't know. I'm assuming they're factoring in like what kind of computing the human brain is doing, like what kind of computing the computers are doing. I'm assuming there's context somewhere. Although I guess like human brains don't really like vary that much in energy consumption, even if they are doing more difficult tasks, I suppose. Anyways, that's the end of my notes for today. I'll be back tomorrow with more. All right, let's get started. Day 4. They coined the term organoid intelligence. Apparently organoids can contain myelinated axons, which is a little bit surprising because I think we were learning before that like normally myelinated axons are going like across large distances, and these brain organoids are not large. And these organoids also do things like show spontaneous electrophysiological activity, which is cool. And they also demonstrate complex oscillatory behavior, high cell density, layering patterns, all these little things that make them more and more like real brains and not just like the little 2D layering cultures that they were using. They make a lot of big claims about what these little organoids can do. Unlock new AI algorithms, overcome current AI limitations, and develop new brain-computer interface technology. So apparently they've been doing this kind of thing for 50 years, or the concept at least. There was a lot of work they did on animals in the early days. They started out using the lamprey. They used a couple of different species to study like the cellular level of what's going on here. When they were doing it in rats, the neurons were like self-organizing and were responding to stimulus like electrical information. They were self-organizing. There's a bunch of different stuff that seemed promising when they first started. And then they said something that was a little bit strange and kind of clued me into like, maybe I'm not getting the full story here. To the best of our knowledge, no relevant approach using brain organoids as learning systems has been reported. And I was like, what? Because for me as a person, having done no research, like that first article we read talked about like the brain organoids playing Pong. That seems like something has learned something, which is funny because they said that and then immediately go on to list 2 different studies that are yielding results of, or like kind of studying what they were wanting to. It's like, why would you say there's none and then list 2 immediately after? This one, the recent work by Kagan et al. And a study by the European Union. So at this point I was kind of like, wait, why are they saying that when I'm pretty sure there's lots of other research that has been done on this? So I started kind of skipping around, and then I got to where they said in vitro experiments have demonstrated the ability of these cultures to both control robotic systems and play simulated video games. I was like, wait, why would you not add more about that? What's happening here? And then I realized that because this article was posted in February of 2023, all of those other sources that I was looking at have been posted like 2 or 3 months ago. And I'm currently in May 2025. So this article is pretty far out of date, which is a bummer. Anyway, so I looked around a little bit more in this. I was looking at the figures. Here's one. Let's see if I can show this whole thing. So they've got like actual figures about what they're doing when they are studying these things. So this is like you combining AI machine learning with big data management to kind of govern this whole thing. So the inputs they've got are like electrical signals put into the organoids. They're using chemicals. They're using optogenetics to kind of look at or to kind of give input to the brain organoids to send messages to them. And they're also using like retinal organoids or combining multiple to like get better answers. So I guess if you connected a retinal organoid with a brain organoid, it would be able to give that brain organoid like input, I think is what they're trying to say. And then the outputs they're using to measure these things are like electrophysiological recordings, imaging. They're also using fluorescence. I'm not sure how. And confocal microscopy. And then here you've got the little organoid with these little MEA measuring scopes, like kind of wrapping around it. And then you've got a probe inserted into it, which is kind of nice to know. So then I got a little sidetracked and I wanted to know how many neurons a brain organoid has, because the brain has like 100 billion, right? So if you've got tiny little ones, how many does that have? And I came here to the Wikipedia page and I saw this picture, which is kind of cool. is like actually the organoids in there. And those are a lot bigger than I thought they were going to be. Those are like the centimeter sized ones. But then I looked around and I didn't see any like numbers about how many neurons organoids have, which they probably haven't measured this because it's kind of a random thing. So it's like, okay, fine, that's whatever. I don't need to know that. I was also wondering about like glial cells and how they're working in these organoids. Like, do they have glial cells? They probably do. What else is going on? Like, we learned all this stuff about the actual brain and neurons and how they work. Like, do the organoids have all those things? How can we find out? But I decided it wasn't super worth it to look into because I'm probably not going to find any sources that speak to that kind of thing directly. So I came back here to this article and let's look at more figures. I found another cool figure here. Here you've got the 2D neural culture, which is like not that great. And then you've got the 3D one, which there's a whole bunch more going on here. And then you've got, so they do actually show here, you've got your other glial cells, the astrocytes. This one I'm not going to try and pronounce, and the microglia. So the red one, the blue one, and the green one are all glia, and the pink one are the actual neurons. And then down here you've got this cool map of like, you can actually see them growing, I think. So in this key, the pink are neurons, the red, they're kind of matching up with this key up here. So the green are astrocytes and the blue are nuclei. See if we can get wow, that was really close. Let's get a closer picture. So here's like the early days, week 4. So you can see the pink are the neurons. Doesn't really look like there's much else other than just the neuron and the cells. I'm not sure. Maybe these blue things are the nuclei. And then this is week 8. You can see the astrocytes starting to come in there. This is week 10. You've got more astrocytes, more neurons, the nucleus. What else we got in there? And the red are the long word things, week 12 and week 15, which is getting kind of, the astrocytes are kind of taking over. It looks like where are the neurons even at week 15? Astrocytes are like all over the place. And the nuclei are getting real big. And this is the big old organoid at week 15. Look at the axon going all the way across. That's kind of cool. That one's myelinated for sure. So at this point, they say a couple interesting things. Say that most of the brain organoids show spontaneous electrophysiological activity and are reacting to electrical stimulus, which is cool, which is necessary for computers. Turning them into computers, they say that the oscillation waves are comparable to like the waves from human preterm babies. So it's like baby brains that are not fully developed yet. It's kind of showing like similar to how these brains are working. Axons in these organoids are like super are showing extensive myelination, which is surprising. Like I was saying before, why are they getting so why is there so much myelin? And as the myelination increases As myelination increases electrical conductivity approximately 100-fold. So they're saying that as the myelination increases, it's like going to boost biological computing performance by a lot, by 100-fold. I'm wondering if there's something they're doing to encourage myelination, because I thought it was only long axons that got myelinated. Let's look at this figure too. So here they've got like the standard EEG, like how they measure brain activity. So I think they're just showing that they're taking like something similar to this and they're like super condensing it into this tiny little thing. So this blue thing is basically that blue thing that wraps around your head, and that's how they're using it to measure data, measure electrical activity. And then here's like a picture of like the actual organoid with the EEG thing going around it. I can't tell if these are like probes being stuck in or if this is just part of the EEG process. Here's another little view, 500 UM. I don't know what that unit of measurement, I don't even know what that letter is. And then they're just showing how different channels produce different signals when they're measuring the organoids. This is showing the voltage over time. Not super useful, but it's kind of interesting to look at how they're measuring these things. Oh, so these are each of the channels. So like channel 1, 2, 3. So channel 1, 2, and 3 over there. All right, so at this point I was like, let's look for some more current information because this article is pretty far outdated. It's 2 years out of date, and there's been a lot of other work that has been done to further this research, especially the stuff that I like initially started looking at when I was looking at this. I watched several YouTube videos, and they were all talking about a bunch of different stuff like brain organoids learning to fly as butterflies and learning to play Pong, and like they're like putting them in dishes and like linking them up with the axons. And I was like, where's all that information? So I went looking for that. So I came here because this expanse talk was where I got one of the more interesting videos. Key takeaways from the expanse. So I looked through this and this was absolutely useless. So I guess the expanse is kind of like a sort of TED talk, like a summit for like innovators. And they were talking about a bunch of different stuff. So this just lists all this different stuff they were talking about at expanse, which is not helpful. So I came back and went here, and I didn't get a chance to read this yet, but then I was looking at the video that talked about like the axons in the two different dishes where they were connecting and like learning to fly as a butterfly. This was the video that talked about it, and I was kind of going through it, but I didn't get a chance to take any notes. Anyways, that's all for today. I'll be back tomorrow with maybe the last bit of info. Home stretch, here we go. Okay, so he cites a couple sources in this video. The first one is the assembly of functionally integrated human forebrain spheroids. So let's look at that one real fast. Here it is. And so just as I was reading the abstract, I realized that this doesn't really have anything to do with organoid intelligence or organoid computing. This is a study that's looking into how the different parts of the brain are like crossing the different brain regions to like talk to each other. So the axons are, he calls it a handshake in the video. The axons are spreading out to kind of link together and form a bridge that will go between like the hippocampus and the left hemisphere and the right hemisphere, so that all of the different regions of the brain are talking together. And this is what that study is doing, but they're using organoids to talk about it. Although they say spheroids in this publication, and I'm not sure why, because we learned earlier that spheroids and organoids were different. But so I kind of came back to this video and I kept watching a little bit. So here you can see that the different regions of the brain are like reaching out, and the axons are getting super long to complete like this neural handshake. And here's where they put them on like a microchip, and they put the different organoids in like the different little parts of the thing of the microchip. So they've got a little, you can see there's a little pathway that's going between them. And then they started like reaching out to like link together using axons. So the axons are reaching out to kind of link together. And then they started syncing up, like all of the different bursts of electrical energy were linking up in the spheroids. He makes the point that this bundle down here that's like connected by the two axons is faster at communicating than two organoids that were just put right next to each other and kind of fused together. Although I'm not really sure how they're fusing together. Would it not be also that they're fusing together via axons? Also, how are the axons fusing together? Because axons don't touch dendrites unless something else is going on here where the axons are actually connecting. I'm not sure. Maybe they're like the double-ended axons. I can't remember what those neurons were called. But anyways, so they tested these, like cutting the neurons to see what happened to make sure that they were actually syncing up, and that's what was actually happening. And then they shone a light on there to test that. I feel like I'm bastardizing this guy's video. Sorry, guy. It was a good video if you're interested. But by the time I got to the end, I realized that this has literally nothing to do with organoid intelligence, except for the very last like 5 seconds he mentions it. It's all about neurons and how they link together. And then I remembered, oh, right, I actually watched these videos out of order, and then I put them in order in my notes so they would be chronological. But let's watch the other one where he talks about actual organoid intelligence. So this is the one where he talks about like the little butterflies being flown by the organoids that are hooked up. And the company that is doing this is called FinalSpark. FinalSpark.com they are the ones that are trying to figure this out, and they're the ones that had the little butterfly game, although I didn't see it anywhere. So you can control this. They're using like crowdsourced data input, so you can like log onto their website and control this butterfly from anywhere, and that will help to train the brain organoids. So you can see them in this little chip. So if you go to live view on FinalSpark's website, you can see the little organoid there, and it's got all these little electrodes connecting it, and you can change the number of organoids, I guess. I'm not sure what this MEA means, but there's like 4, it looks like four connected on this one, even though it says 3. I see 4, and this one has 4 as well. This looks like the image from here. So you can see they've got the little like lines, electrode lines going up to the organoids and connecting. They've got like little ways to measure it. So apparently they've grown them real tiny. These ones are only 0.5 millimeters. This is a cool video of the like the neurons interacting. Like it's sped up obviously, but you can see all the neurons like firing and the electrical stuff going on there. I'm trying to point with my finger, which is pretty cool. So I'm not sure where he got these videos from, but here's like a little video of like the actual stuff in the cooler, where they're like keeping the stuff at the right temperature and like putting the inputs in. Anyways, and here you can see like they've got the little electrical stuff. I think that looks simulated to me. I don't think that's actually what's going on, but this is essentially what they're doing is they're using these electrodes to zap the organoids, and like that electricity is obviously simulated, but they're using the electricity to like stimulate the organoids and then measure the responses. So they're like stimulate, measure, and they're that way they can train the organoids to do things. So, and then this was the video about the guy in The Expanse. He makes a bunch of points that we've already covered. He talks about why they're doing it, and like humans have intuition, AI doesn't. If AI was lost in the jungle, it would get eaten. The AI can pretty much just copy what humans are doing. It can't like create itself. I don't know. Pretty much all of the points he made were stuff that we had already covered. There's that image from that article we were looking at where they've got the organoid hooked up to the electrodes or whatever, the EEG machine. He said that they're using these little games like they're like Pong game and all these different things to train the organoids and like little robotic stuff, but I didn't have time to look into it. And then he started talking about how like the ethical concerns, which came up in pretty much all of the articles that I was looking at, the ethics of this, like can the organoids become conscious and what happens if they do become conscious? At this point, they're basically like so tiny that they're just neurons and there's no possibility of context, like not even close. But I guess it's a concern. He said there's like an ethics team working with them. And what I really want to know is what the heck does that look like? Like, what is the ethics people doing? Are they like, you can't do this? Are they telling them what to do? Are they being like, you've crossed the line? Or I'm not sure, honestly. I would like to know. Anyways, this video didn't really help me out much. And at this point, I was pretty tired of not this topic, but like the scope, the amount of information that I would need to know to like really dig more into this topic was just very overwhelming because there's just so much. And I kind of decided that I think I'm okay with that being all that there is. So thanks for hanging out, learning stuff about organoids and brains with me, and I will catch you in the next one. Peace.